Hello and welcome to another episode of the Omics Cast. Today we're going to talk about polygenic risk scores. So this episode of the Omics Cast is in response or reacting to a tweet by Ewan Burney, the co-director of the European Bioinformatics Institute, the EMBL outstation in Kingston in Cambridge in the UK, which is him tweeting and writing a blog post on something that has happened recently, which is a company in the US has started selling tests to do embryo selection for essentially intelligence, educational attainment. And so the blog post by Ewan Burney starts by saying about this American company that's offering a polygenic trait score on embryos, including intelligence, and how Ewan Burney thinks that this is wrong on a number of levels, and he goes on and talks about this. The first one is that there's no way something like this would be safe and effective for embryo selection. And the second is that IQ, intelligence score or educational attainment trait, is a trait that's so complex that this wouldn't work anyway. And there's some plots here to show this later. First of all, ethics. And Ewan Burney goes and describes what the ethics of these are like and what other examples we have that are similar. He says, even if science progresses and allows us to do something, it doesn't mean that as a society we should perform these actions if we collectively as a society consider it wrong. And the example here, and I think that's very understandable to anyone, is the fact that we have the technology to do ultrasound scanning of fetuses to reliably identify what sex they are, if they are males or females, if they're going to be boys or girls when they are born. And you can do this early on in pregnancy. However, almost everywhere it's explicitly illegal or implicitly prohibited to select the sex of your child and to go for abortion, even if you don't like the sex of the fetus. It says similarly, there are ethics considerations for embryo selection, meaning if you have a bunch of embryos in a petri dish, there are ethical considerations on selecting those embryos based on something as difficult to measure as intelligence. And then he talks about the science side of polygenic scores and says, even though he's a genomic scientist and he's cautiously enthusiastic about using genetics of polygenic traits, this, he says, would potentially augment our understanding of risks for common diseases in adults. And common diseases here, they read things like cardiovascular disease, diabetes, etc., etc. So things that most of us will end up suffering to some degree during our life. What can we do in terms of the genetics and the genomics research to better understand this and better treat it, right? And he says for embryo selection, then these polygenic scores don't apply, in the sense that there's a very high potential to cause harm and only small potential to mitigate bad outcomes. So what is a polygenic score? A polygenic score, according to Wikipedia, or polygenic risk score or genetic risk score, is a number that's based on the variation on multiple genetic loci, multiple genes, and their associated weights with respect to a trait or to a disease, right? So the way Ewan Burney explains it is, a polygenic traits are traits where multiple places in the genome contribute to a trait. Poly meaning many and genic meaning genes, right? And he says this is all fine. We can calculate this with more and more precision lately, but it's not the whole picture, especially for things that have to have a very strong developmental aspect. It says for some traits there are strong developmental aspects, which mean that the polygenic score we select on also contributes to developmental effects and other features in adult life that are very difficult to anticipate, right? Some commentators cite the success of animal breeding schemes, and then Ewan Burney makes a point, and we'll see some tweets about this later on, that these schemes are not the same as embryo selection, in the sense that animal breeding, genetics, gets its main benefit from selecting mates for breeding, not from selecting embryos of a random mating process, right? So even if you can sperm and eggs to the gametes to produce embryos in a petri dish, that selection of embryos is not the same as selecting the parents, selecting the mates that would be producing those embryos. And he says anyone who's using plant or animal breeding as a justification for the for success of its genetic intervention in humans, then he argues does not understand animal breeding. So if you want to get better cows that produce better milk or better meat, better beef, etc., you select the parents, you select the cows that you're going to be breeding. Whereas if an American company is trying to sell snake oil here for a couple of future parents to tell them, we can use your gametes to produce a bunch of embryos in a petri dish and select them, the parents have already been chosen because there are the customers. It's different than choosing the parents and then basically saying the best parents for a specific trait would be those two. He also says this is not about genome editing. The amazing ability to directly edit genomes is of no relevance to this discussion. A polygenic trait prediction should perhaps be better stated as an interpolation, or trying to estimate a phenotype based on all the background information, all the reference data we've accumulated from the many previous individual phenotype and genotypes. And he says the most powerful methods do not to do this definitely do not model any specific base pair change due to statistics and issues that are behind, and that there is blocky nature of human genetics that impedes us from knowing what will happen if we change one single base in the human genome, right? So let's not muddle the topic here and not talk about genome editing, CRISPR-Cas in the context of what this American company is trying to do. And then Ewan Burney, again, the author of this blog post, goes and talk about intelligence and educational attainment as a polygenic trait. He says the genetics of intelligence and of educational attainment, how well people do at school, is a very complex topic. Nevertheless, some real progress has been made, in particular over the last 10 years. And he suggests a list of authors and people that you can follow in online to get on to understand this complex field better. Stuart Ritchie, Paige Harden, Alex Young, Ian Dreary, Robert Plumin. He says the genetics of IQ and educational attainment are real polygenic traits, but they are the sort of traits where one should be very particularly careful of thinking about embryo selection. Over and above the genetic concern, and he in this blog post makes a comment about when people first started selecting chicken, they realized they were selecting for one trait where they ended up selecting chicken that were very good at fighting against each other, but not breeding together in a chicken coop. And so that genetic concern is on top of that. Even then, there are more issues with using polygenic risk scores, PRSs, or polygenic scores, PSs, for embryo selection of humans. And he says even the best methods will probably give us very little in return. We're trying to select for or avoid severe intellectual disabilities. And he says there are a number of scientific reasons why this is the case. There's two of them. The first one is that it's a non-random environment, meaning once you put a kid through schooling, a bunch of other things have already happened that are not random, as in what happened randomly in a petri dish. The issue of localized genetics, cryptic population stratification are to be considered. Second point is that one-third to half of the genetic signal for intelligence in educational attainment is due to parental environment, right? And so what's the point of choosing these, which, commercially sounds like a very interesting proposition, will make your kid clever by selecting embryos from a petri dish, if actually you cannot do that because quantitatively the genetics don't add up too much. And we'll see a couple of slides relating to this later. Estimating the tails has an additional major complication in trying to isolate the true within individual effect, meaning you have to end up choosing one embryo of that petri dish. That doesn't mean that you're selecting the best one. So this American company, snake hole position, how can you rank or significantly select for the top end of a continuous scale? Main determinants of every embryo selection will be the genetics of the father and the mother. One is bounded by these genetics, formally a small variation around the mean of father and mother in the models used. In practice, rarely goes outside of this expectation. So the two sides of the coin here is the father and the mother have already decided they're going to have kids. Compared to animal breeding, you've taken most of the power out here. In summary, Ewan Burney, director of the European Bioinformatics Institute, EMBL, says then what should not use embryo selection for complex trait behaviors. I think I agree with him 100%, and it would be scientifically unsound to do so for any trait, in particular for IQ or educational attainment traits. And then has posted this blog post on it and on Twitter about it. He got some answers about this. One around animal breeding, saying, oh no, animal breeding schemes do not involve embryo selection. They actually do. He says, in a way they do, or they may not do, but the main argument here is that you select for the mates, the two animals that you want them to breed, right? Luke Coleman, senior lecturer at the University of Melbourne, who writes papers about sexual selection for improving fitness, etc., comments on this and says, I think embryo selection is effectively being used to select on long-lived plants, trees grown for their oil content. Selecting on adult trees takes too long, but one can grow a bunch of seedlings, embryos, in quotes, you should say here, genotype them, pick the ones your polygenic score recommends. Ewan Burney replies, I still think the trait selection in trees is done by selecting good parents, certainly not selecting from wild, of course, but I can see that the genotyping of young from a cross might help less than choosing good parents. And that this is not embryo selection in the sense that the seedlings are not embryos anymore. Luke replies, true, it's not embryo since the trees are at least germinated, but if that method works, then embryo selection should too, provided we could effectively measure PGS for desired multivariate trait combination in humans, which we can't. And so the agreement here is that indeed what the American company is trying to do is snake oil. They're trying to say we're going to make your kid clever by selecting embryos from a petri dish. Ewan Burney makes a good point here, made many years ago, which is a young plant has gone through critical development phases, an embryo hasn't. It's not birth, marriage, or death, but gastrulation, meaning the development phase, which is truly the most important time in your life. Many mammalian embryos fail in the gastrulation to implantation phase, and all the critical development happens very early in, with small differences, potentially fixed for the lifetime. Thought-provoking seems to me it be possible to calculate the expected response to embryonic selection by polygenic score. Do you know of any potential theoretical work in this area? And then Ewan mentions this paper, Screening Human embryos for Polygenic Traits, has limited potential. And it's a paper that's been published recently, relatively recently, May 5, 2019. Leonid Krugliak, also very active on Twitter, and a bunch of other authors. And they say polygenic score PS predictors can explain increasing proportions of variance, but can you use them to screen human embryos? They say that they see the gain increases very slowly with the number of embryos, but more rapidly with increased variance explained by PS. Average gain due to selection would be for two traits here, 2.5 centimeters if selecting for height and 2.5 IQ, intelligence quotient, if selecting for cognitive function, right? So it's good that they do both here, height and IQ, and both measures are, numbers that we can understand. We can all understand what 2.5 centimeters are, a bit less than an inch, and 2.5 IQ points, intelligence quotient points, also a very small amount. And there's a fantastic plot. It's very easy to understand because of the colors are so clear in this paper, which is analysis of selection for height in 28 real families that had 20 adult offspring each, right? So these are large families where they had lots of kids, and then they measure, in this case, the plot is for height in centimeters. They measure the height of the kids, and then they looked at the top predicted PRS or PS individual was in red, and then they look at the outcome, the result of the height of the offspring in gray, whereas the parents are here in blue. And they show that it's usually not the actual tallest sibling, the one that gets the highest prediction, the tallest prediction in red. So for example, here, the red one is on the high side, but look at what happens in this second family here. The red, the one that would have been selected on an embryo selection on a Petri dish, is actually 166 and 67 centimeters, where both parents are at around 175, and two of the siblings are above the parents, one at being 183. So this for height means there's a non-insignificant chance of this American company selecting for a kid that's going to end up being 166 centimeters tall, where naturally both parents, they could have had a kid that was 183 centimeters tall if they had not done nothing else, right? And so look at all the blue versus red and gray here, and how it's almost never then the red box is the best actual selected individual for this trait. So if you like this podcast, if you like this video cast, I recommend you to follow me on Twitter, twitter.com/my name and surname, Albert Vilella. I'm happy for you to also contact me via LinkedIn and ask me questions. You can follow me on Twitter, and we can DM if you have any questions as well. LinkedIn probably even better for that. I contribute to a podcast, an audio cast called Cambridge Genomics Precision Medicine Cambridge, with Manuel Corpus and Vijaya Parthivan as well. And we've already discussed about polygenic risk scores in a couple of episodes. I recommend you to have a look at it. And this is it for this episode. If you want to learn more about this, subscribe to the channel. I'm sure I'm going to be talking more about polygenic scores in the future. Until the next episode, goodbye.